This video covers Higher Human Biology, Unit 3, Key Area 4, the cells of the nervous system and neurotransmitters at synapses. The key areas and outcomes we will go over are describing the structure and function of the parts of the neuron, explaining how myelination changes throughout childhood, giving the stages of nerve transmission at synapse, describing how a threshold can be reached through summation, giving the function of endorphins and dopamine, and giving examples of the involvement of agonists and antagonists in nerve transmission. So this is our simple diagram of the neuron. We're going to build it up as we go. You'll notice in your summary booklet when you see it next week that it's a bit more of a complicated picture. So we start with the cell body. This contains the nucleus and most of the cytoplasm. It's the control center of the cell in terms of metabolism and contains clusters of ribosomes which produce proteins such as enzymes needed to make neurotransmitters. We can see that this here is the cell body, and on the outside we have things called dendrites. Information from other nerve fiber cells will come in through the dendrites and be received in the cell body. This information is processed and then sent along what we call the axon, which is this nerve fiber which runs away from the cell body. So the direction of nerve transmission is from the cell body along the axon. The axon is by far the largest part of the nerve fiber. If we imagine something like a giraffe, they can actually have axons which are 15 feet long, which is very, very long. And things like the sauropod dinosaurs could have ones that were up to 50 feet long from the tip of the tail and to the brain. An important part surrounding the axon is the myelin sheath. So myelination is a process that occurs from infant age through early to late childhood. The myelin sheath is a fatty layer that covers the outside of the axon. It provides insulation and increases the speed of impulse conduction. So I like to think of the myelin sheath like the insulating plasticky substance on the outside of electrical wires. Without the myelin sheath, the signal would get diffused and wouldn't reach its final site. So as stated, when we're born, we don't have an awful lot of myelin around our axon. So you can see the cell body with the dendrites, and then leaving it, we have the axon. Throughout childhood development, we go through the process of myelination. This is carried out by what are called glial cells. These cells also support the neuron throughout their entire lifespan. For this reason, stimuli in the first 2 or 3 years of life are not as rapid or coordinated as those for older children or adults. So here, the signal in toddlers, for example, would get diffused before it reached its endpoint. So toddlers tend to be quite clumsy, even through kind of early to late teenage years. A little bit of clumsiness should be expected because the axon hasn't been fully myelinated. As you see in adult and late adult stages, we can juggle 4 or 5 frisbees at the same time and play a concert grand piano. So we have the cell body with the dendrites, we have the axon covered by a myelin sheath, we have the direction of nerve transmission, and then at the very end of the axon, we have a stop. So there's a gap between the end of the axon and the dendrites of the next nerve fiber. This gap is what we call the synapse. And as stated before, it's neurotransmitters that are able to carry the signal from the axon across the synapse to the next dendrite cell. So in your summary booklet, again next week, you'll see a diagram that looks like this. We have the direction of nerve transmission, and it reaches this endpoint here that we can see in the diagram. Neurons are connected with other neurons at the synaptic cleft. Neurotransmitters relay impulses across the synaptic cleft in the form of chemical transmission. So before nerve transmission, neurotransmitters are stored in vesicles in the axon ending. So the electrical signals come along, it stimulates the vesicles to move down, and fuse with the cell membrane at the end of what we call the presynaptic neuron. Because this is synapse, so this is the one before it, the presynaptic neuron, and the neuron afterwards would be the postsynaptic neuron. These vesicles have been stimulated by the electrical impulse to fuse and to release their neurotransmitters, which is what they're filled with, these little dots. These neurotransmitters diffuse across the synapse, this gap, and they will fuse to receptors on the postsynaptic neuron. You can see a beautiful animation of this. So coming from the presynaptic neuron to the postsynaptic neuron. We have the receptor here, and as with all proteins, the receptor is specific to only the neurotransmitter that it has been designed to work with. You can imagine that if a neurotransmitter has been released because of a signal transmission and fuses to the receptor, it will stimulate potentially a nerve impulse to carry on in this postsynaptic neuron. What we don't want is this single neurotransmitter to send multiple signals down the postsynaptic neuron, because this means we're going to get a continual stimulation. And if, for example, this was a pain stimulus, one pain impulse, we don't want that to become 1,000 pain impulses on the other side. So we have to remove the neurotransmitter. The two main ways we talk about this happening is by the use of enzymes. So in the postsynaptic neuron, the neurotransmitter can be broken down by enzymes, or it can just be reabsorbed. So it can either be broken down and we can try and form it back into a neurotransmitter, or it can just be detached and reabsorbed. It's actually the decision of this receptor here to decide what function that neurotransmitter is going to carry out. Neurotransmitters have two main functions. They can cause signals to be produced in the postsynaptic neuron due to excitatory signaling, or they can dampen down the signal and reduce the likelihood that it is transmitted. This is what we call inhibitory signaling. It's the receptor in the postsynaptic neuron that decides whether or not the neurotransmitter signal that's sent is one which is going to be excitatory, so encourage more signals to be sent, or one which is going to stop signals being sent. At times, we can have very, very weak stimuli of our senses, so our sense of smell, sight, hearing, touch, taste. And it can be that these stimuli do not release enough neurotransmitters to cross the synapse for nerve transmission to continue at the other side. So a minimum number of neurotransmitter molecules must attach to the receptor in order to reach the threshold on the postsynaptic membrane and actually encourage the transmission of this impulse. If fewer neurotransmitters are received, then the synapse can actually filter out this signal, as it was likely caused by quite a weak stimulus. In convergent neural pathways, which we've already talked about before in 3.1, several sensory neurons can join together, and if each has quite a low transmission of neurotransmitters, they can group through what we call summation, and together release enough neurotransmitters to trigger that impulse. So if this was the light receptors in the eye that have each sent a very, very weak light signal, these can be grouped together and actually achieve enough of a signal to stimulate an image and stimulate electrical transmission. So it's quite important in convergent pathways for this to happen. We talk about two specific types of neurotransmitter in section C. We talk about the endorphins and we talk about dopamine. So they both have quite similar, but importantly different jobs. So endorphins, they stimulate neurons involved in reducing the intensity of pain and up-ramp the feeling of pleasure. You can trigger these if you have a severe injury, prolonged or continuous exercise, if you're quite stressed, certain foods, and sexual stimulation. So we can see in this diagram here, we have a neuron which in its vesicles have the green neurotransmitters. The green neurotransmitters could come across the synapse and fuse here. If we assume these are neurotransmitters for pain, it might be that, yes, your leg is absolutely killing you, you've run 40 miles, but actually your brain's got the message now and it doesn't need any more pain stimuli. But your leg is still sending those stimuli across the synapse. So what your body can do is another neuron can send neurotransmitters across, like endorphins, and this can inhibit. So we talked about excitatory and inhibitory. This can inhibit the release of these neurotransmitters here. So the endorphins would bind to their receptors in the postsynaptic neuron, and that would stop the production, the release even, of these neurotransmitters here. You can see that the receptors the endorphins bind to are called the opioid receptors. Now, you might know opiates can be things like morphine, heroin, diamorphine, things like this, and we'll come back to that in a future section. We can actually produce chemicals which mimic the effect of things like endorphins and kind of misuse this pathway for our own means. So it's switched off the pain transmission in this network. Dopamines induce the feeling of pleasure and reinforce particular behaviors by activating a reward pathway. So the reward pathway is activated when an individual engages in a behavior that's beneficial to them. So for example, eating when we are hungry. So it might be that when you're very, very hungry, you eat something and you feel absolutely amazing. That's not just the sugar rush, that's also the dopamines that your body is producing. It's trying to encourage you to eat when you're hungry because this is obviously very useful. We can also release dopamines when we see people liking our pictures on InstaChat or Snapagram or any of these other non-copyrighted social media apps. And this again will reinforce our dopamine pathway. It might be that in the antiquity of evolution that social interactions were very useful in our behaviors. So we've still got the mechanisms through dopamine to reinforce those kind of behaviors. So antagonists and agonists. We're going to talk about two different types of neurotransmitter and drug and how they affect the neuron. So normally a neurotransmitter binds to the receptor on the postsynaptic neuron. Hopefully I've said that enough times now that we recognize that statement. So we've talked about the fact that this can either cause excitation, so it can encourage signals, or can inhibit them, like we had with the endorphins here. So this is just the normal pathway. The neurotransmitter binds, goes across the postsynaptic neuron, and it does its effect. We can have chemicals such as agonists, which bind to a specific receptor on the postsynaptic neuron, and that stops the neurotransmitter being able to bind. When we talk about competitive inhibition in a further part of the course, it's a similar mechanism to this, although that's potentially more similar to antagonism. So this chemical here, the orange one, has bound in the place of the neurotransmitter, and there is no space for the neurotransmitter to bind. But this is okay. In an agonist's case, you get the same reaction you would have before. If the neurotransmitter was supposed to encourage a signal, the agonist encourages the signal. If it was supposed to cause inhibition, the agonist causes inhibition. So this is fine. It's doing the job of our neurotransmitters. In the case of an antagonist, however, this is not the case. So an antagonist will bind to that receptor, and again will not let the neurotransmitter bind in its place, but it does not have an effect on that receptor that the neurotransmitter would have. So if the neurotransmitter would have caused its excitation, the antagonist causes no effect at all. So you're missing out on that excitation. If it would have caused inhibition, again, nothing. This can cause a lot of interesting effects. So if we think about antagonism, this that we've just looked at, the antagonist in this case, the green one, has bound in the place of the neurotransmitter. This means that the neurotransmitter cannot get into its binding site. no effect has been received by the neuron because the antagonist does not encourage the same effect as the neurotransmitter. This means that your body tries to fix this problem and it produces more receptors. So that means its neurotransmitters now do have somewhere to bind. What this can mean is we get sensitization. So whereas normally on a postsynaptic neuron you would have 2 receptors, and this means that if your body produced 100 neurotransmitters, there was still only a reasonable number of receptor sites for it to bind. So if you had 100 neurotransmitters, you couldn't get a massive effect because there are only 2 places for the neurotransmitter to bind. But because you've maybe started taking a drug that mimics the effect of the neurotransmitter, your body doesn't really understand what's happening, so it produces more receptor sites. This means that you're now overly sensitive to this drug. You now have a lot more places for it to bind, and you get what's called sensitization. It leads to addiction, where the individual can crave more of the drug. We also get an effect from agonism. So you can get drug tolerance. This is some agonists, for example, are opiates, so which we talked about with endorphins, things like heroin, morphine, diamorphine, OxyContin, things like that. So agonists stimulate the body's receptors at the postsynaptic neuron. And because the effect is being received without the body needing to produce its own neurotransmitters, it down-regulates the sensitivity and number of receptors it needs for that particular neurotransmitter. So it decreases how many neurotransmitters it actually has for that, how many receptors it has for that neurotransmitter. Desensitization can lead to drug tolerance, where the individual must take more of the drug to get an effect. Because if you imagine, if you start taking a lot of these drugs, your body will drastically reduce the sensitivity of these receptors and how many there are. So you have to constantly be taking the drug just to get your normal effect. So we've discussed the structure and functions of parts of the neuron. We have talked about dendrites, we've talked about the cell body, we've talked about axons, and we've talked about the myelin sheath. Hopefully you know the structures, where they are, you could annotate a diagram, and you know the functions of these parts. We've talked about myelination, and the fact that when we're born, we have quite a low level of myelination on our axons, and it grows through development. This is why quite often there's less coordination in the actions of infants and toddlers. There are some diseases that also can decrease myelination, and this obviously can be bad. We've talked about the fact that glial cells are the ones that produce the myelin sheaths, and that they also support the neuron through its life. We've talked about the role of vesicles in storing neurotransmitters at the presynaptic neuron. We've talked about the synaptic cleft, which is the endpoints either side of the synapse. We've talked about the receptors in the postsynaptic cleft quite a lot. We've talked about why it's important that neurotransmitters are degraded by enzymes or taken up and reabsorbed into the presynaptic neuron after they have been received by the receptors. We talked about the fact that signals can be excitatory or inhibitory, and what that means with examples of the pain pathway and why we might want to inhibit that using endorphins. We talked about thresholds, so weak stimuli may not produce enough neurotransmitters to hit the threshold, so they might be summed together through summation, and the fact that this quite often happens in our convergent neural pathways, such as the rhodopsin pathway in light. We've talked about endorphins, and we've talked about dopamines. We've talked about when they get released and what they do. Endorphins being part of the damping down pain through the opioid receptors, and the dopamines being involved in rewarding us for behaviors that are good. We talked about neurotransmitters acting as agonists, where they occupy the receptor site instead of the neurotransmitter. And we've talked about antagonism as well, where a chemical occupies the receptor site but does not cause the signal generation through either excitation or inhibition. Finally, we talked about the fact that because of agonism and antagonism, the body can adapt. So in an agonist, it will become tolerant because it will reduce the number of receptor sites it has and the sensitivity of these receptors. And in antagonism, we get sensitization. So because we have blocked the receptor site for the neurotransmitters, it increases the number and sensitivity of these receptors. Hopefully that made sense. If there's anything that you don't understand, please do ask your teacher for help.